In developmental biology, we learn different concepts on the development and growth of different individuals. What if we are able to regenerate animals' limbs because of these advancements in knowledge, especially in developmental biology? Hello, and today we are going to present to you a strategic plan on how we can help restore vivor's limbs. But first, who is vivor? Vivor is the mammal of the species Macaca fascicularis, or also known as the long-tailed macaque. Vivor was born with a special condition wherein Vivor has incomplete limbs, specifically arms. Vivor specifically suffers from terminal transverse limb deficiency, where the development of Vivor's limbs was interrupted, thus Vivor missing a portion of his arms and his hands completely. Though Viver himself cannot regenerate his limbs naturally due to restrictions brought forth by his species, certain organisms are capable of doing so. Amphibians such as salamanders have been reported to have the ability to regenerate their body parts. In this strategic plan, we aim to restore the limbs of Viver using the same regeneration mechanisms of salamanders. Specifically, these are the steps to be taken: wound closure, formation of the growth and plastema, and growth and morphogenesis. The first step for Viver's recovery is wound healing. As a stump and scars are present in Viver's forelimb, cells are sealed off for us to work with. As such, creation of a new wound must be done on the site. The presence of a wound induces the migration of epidermal cells to the site to seal it and form the wound epidermis that produces the chemoattractant STF1 needed later in the process. This runs smoothly in amphibians as the entire thing takes only 24 hours. However, in the case of mammals, migration of epidermal cells take too long, thereby delaying the formation of the wound epidermis and reducing regeneration abilities. Hence, it is imperative to find a way to hasten the migration of epithelial cells. This can be done via cell harvesting and culture. Stem cells from viver's bone marrow can be harvested, cultured in a medium, and induced to differentiate into epithelial cells. These epithelial cells can then be introduced near the wound area to develop the wound epidermis. Following the AER or AEC in salamanders, fibroblasts of the connective tissue meshwork break free and migrate at the center of the wound. Fibroblasts proliferate there to form the blastema. In mammals, fibroblasts form scars as a result of the production of an organized network of extracellular matrix material. Fibroblasts of salamanders stop proliferating once normal architecture is restored. As mentioned before, wound induction would be done in order to initiate the formation of the blastema. The blastema, are also known as a regeneration bud, is an important factor in the regeneration of all animals, including mammals. Blastema formation needs to be initiated in order to regrow the severed limb successfully. Although in mammals, this works quite differently compared to our salamanders, since the formation of the blastema and the formation of epidermal cells is much more slower. To aid the formation of the plastema cells and the overall regeneration of revivor's limbs, we need to induce the presence of stromal cell-derived factor 1, or also known as SDF1. SDF1 is responsible for the initiation of osteogenic tissue formation, or in simpler terms, the regeneration of bone tissue and connected vessels. The transforming growth factor beta, or TFG beta, is also needed in order to induce skin formation and overall regeneration of limb tissue. With these induction techniques, we eventually aim to form the plastema in order to induce limb regeneration. The successful formation of the plastema results in cell growth and later on morphogenesis. To promote these processes, BMP2 will be applied to the wound closure. This can be done by loading the cell growth factor in a vehicle and grafting it near the site. BMP2 functions as a morphogenetic agent. The subsequent release of BMP in this area signals nearby cells to multiply and differentiate into bone, nerves, blood vessels, and etc. based upon the positional memory they retain, thereby forming the regenerated limb. As we apply these different concepts on formulating a strategic plan for Vivor, it hopes to give a chance for Vivor to live with full limbs and full life. Here are the references used. This is Estavas and Telen signing